So I was telling that in this lecture practical, we will talk about agarose gel electrophoresis. So going straight to agarose gel electrophoresis, beforehand I would like to tell you about gel electrophoresis. So when I talk about gel electrophoresis, it is a very broad term and can be used for many different purposes. Now, yeah. So now in gel electrophoresis can be used for separation of DNA, protein, as well as RNA. So, like in a very brief or nutshell, if I talk about gel electrophoresis, then I should tell you that it is a process or a molecular technique to separate samples, basically DNA and protein, based on the charge and the molecular weight or size in presence of electric field and a gel matrix. Now, when we use the gel for separating DNA from a mixture of DNA samples, then the type of gel we use is agarose. Whereas if we need to separate proteins from a sample of proteins, then the type of gel we use is acrylamide or polyacrylamide. So polyacrylamide is simply acrylamide, many acrylamide fused together by hydrogen bond. Now from this we can conclude that two different gels are utilized to separate two different type of samples, that is DNA and protein. But the principle of the separation remains the same. So now let's discuss particularly
Ma'am,
about,
in the
sorry,
last line,
yeah,
can over to.
yeah.
Yeah, ma'am. So, ma'am, acrylamide is also used. I like acrylamide is, I think, carcinogenic. So, like, if this is also used, or always the polyacrylamide gels are used. Like polyacrylamide may be more viscous than acrylamide. I guess. I don't know. I'm just
So
asking.
we use polyacrylamide are joined together by hydrogen bond.
Ma'am, I cannot hear you.
Polyacrylamide are joined together by hydrogen bond, they become polyacrylamide. Are you clear about this? Hello?
Yes, ma'am. Polyacrylamide. I understood.
Okay, so we are moving forward. So now let's particularly talk about agarose gel electrophoresis. So this agarose gel electrophoresis, as I told you, is used for the separation of DNA. Now how this DNA is separated is on the basis of size, or we can say molecular weight. Because why we don't use charge at this point? Because all the DNA are negatively charged because of the presence of phosphodiester bond. Okay, now there are different type of DNA of different fragment length. Now they go through the matrix or the gel and they are separated under the presence of current. How they are separated we will study in detail. Here I would like to add one point that the larger molecules, they move very slowly and travels a shorter distance, whereas the smaller particles or smaller fragments of DNA move faster and travels a longer distance. So the separation is basis on the fragment length of the DNA. Now we will take an example and we will see an animated form how it is happening. So first you see the animation, then I would like to elaborate. So what happens, this blue color thing you are looking is a gel, and there are the large sample, medium sample and small samples are written. They are actually the wells. In this wells we load the samples of our DNA fragments and then we uh, apply the current. Now because we know that the DNA are negatively charged, therefore they move to the positive anode. When they move, after some time we just stop the current and we take the fragment under UV and record their image. So this is the basic principle. Now here the similar type of thing is only shown, that a larger particle will travel less where the smaller will travel faster and move to the far distance. Here in the next figure we can see many DNA fragments and they are traveling toward to the gel matrix and there are pores present. Now the pores are of uniform size. So the smaller fragments of DNA could easily move towards the pore and can travel a longer distance. But the DNA fragments which are like longer or having more base pairs couldn't travel that much. So therefore the above bands are of larger fragments and the lower bands are of smaller fragments. Now, where we use this agarose gel electrophoresis. So generally agarose gel electrophoresis are used to estimate the size of the DNA molecule. They are also used to analyze PCR products for molecular diagnosis and genotyping. They are used to determine the quantity and the quality of the DNA and for purification of DNA. Now I would like to tell you that this agarose gel electrophoresis is a very powerful technique to determine the exact length of the DNA. How it is how we determine the fragment length we will be discussing in the later part of the slides. And also they are used to determine the size from 500 base pair to 30,000 base pairs. And the most important thing is that the separation is by due to the electromotive force as the particle passes through the porous gel. Under the influence of the current, they are separated. As I have already discussed with you that the separation of the molecules are basis of size, charge and shape, but in case of agarose gel electrophoresis, it's basically on size. Now, the two most important thing which is needed for the separation of DNA is what the sample we want to separate, and and that depends how the gel is formed. Like if we want to form, if the concentration of the gel is very high, means the agar is present at a very high concentration, then their very fine pores will be formed. So the DNA which have very small fragments are used in this type of gels, whereas if the concentration of agar is between 5, 6, 7%, then they are generally used for larger fragments of DNA. Now let's go through the materials required for agarose gel electrophoresis. So first of all, we require a electrophoresis chamber. You will be able to see everything after I finish the theoretical part. Then we prepare the agarose gel. So what we take 0.8%, normally for the DNA separation 0.8% of agar in 1x TAE buffer. So why we use buffer? So buffer is important to maintain the pH, and in terms the structure of the DNA is also maintained, because we don't want that our DNA get denatured while passing through the agarose gel. Then there is a gel casting tray, then buffer, then staining agent. Generally we use ethidium bromide. Then there is a loading die, a comb and DNA ladder. So why DNA ladder is used, I will also explain this after, like in the later part of the presentation. Then we use DNA samples which need to be like separated. We generally use plasmid. Now we also add loading dye along with the DNA. So generally the loading dye which can be used, or the tracking dyes which can be used, are bromophenol blue, xylene cyanol FF or orange G. They are used to visualize how the DNA is migrating through the gel during the electrophoresis process. And this 1x TAE is the buffer. So how we prepare 1x TAE buffer is by adding 40 millimolar tris acetate, 1 millimolar EDTA. It's a binding agent, binding metal, and we maintain the pH around 8.3. In case the loading dye is xylene cyanol, we add 0.25% of it, 30% of glycerol, and then RNAs. Now, this is the
I
basic
had a
material.
question.
Yes, anybody want to ask something?
Last slide. Last slide
Sorry?
on last slide I had a question.
Yes,
Yeah, ma'am. So what is the like use of ETBR? Standing that so does it has color or something? What is the
yes.
meaning of standing addent here?
Okay, so ETBR is a staining dye which is used to we want to see the DNA bands, right? So how we will see the DNA bands? We will see the DNA band under UV. So for that we need some fluorescence agent so that we can see the bands properly. This ETBR is a fluorescent dye which gives the color, or it adds to the DNA and it's give the fluorescence color when we look under UV. So the bands are properly seen. You will see this in the experimental part, you will better understand it. Okay.
Yes, ma'am.
Okay. Then again, if you have a doubt, please ask.
Yes,
Okay.
ma'am.
Okay, so these are the material required. So as you can see, this is the tray in which we will add this casting tray, and then we will apply the comb, and then we will add the agarose gel to settle. This is totally shown in the form of a presentation in the later part. You will, I think, understand it better in a better way. If you don't understand, please ask me after that. Then this is 1x TAE buffer, which is used both in like while making the agarose shell, also in the electrophoresis chamber. Now what is agarose? So agarose is basically a carbohydrate polymer which is extracted from a seaweed, that is agrobios, and this is basically utilized for the formation of the gel. Now, if I talk about particularly agarose, then agarose, if talked about the structure, it is made up of alternative D-galactose and 3,6 anhydro L-galactose in linked by alpha 1,3 and beta 1,4 glycosidic bond. And you can see in this figure how matrix is formed in the gel. So there are pores and through which our DNA migrate. Now for, as I have already told you that we apply an electric current, so what should be the voltage of it? So because of the voltage, the migration of the DNA takes place from the cathode, that is negative side, to the anode, that is to the positive side. But if we if we provide a higher voltage, then the migration of the DNA will be very fast. But sometime it may happen that the gel melts. So we don't keep the voltage at a very high side. We try to keep it at the optimum. So we shouldn't put the voltage more than 5 volt per centimeter of gel length. Now let's talk about the buffer, that is 1x TAE buffer. Now why we don't use water for like buffer? Simply we can use water in place of buffer. So the thing is, we want conductance. So this buffer has electrolytes because of the salts present, and therefore the conductance is present in this entire method. So during electrophoresis, what happens is water undergoes hydrolysis and breaks down into H plus OH. So buffer prevents the pH change. As a result, the structure of the DNA remain intact as they react with H and OH products. Now the most common type of buffer which we use in the process of electrophoresis or gel electrophoresis is stress. That is stress hydroxymethyl aminomethane. Another compound is added to the stress. They can be either boric or acetic acid, and lastly we add a bind metal, that is ADTA. So we have now talked about two type of buffer. One is TAE buffer and another is TBE buffer. Now what is TAE buffer? So TAE buffer consists of three components, that is stress, acetic acid and EDTA. Whereas TBE buffer contains stress, boric acid and EDTA. Now, as somebody asked me before about the ethidium bromide. So the staining of DNA is done by this ethidium bromide. So what happens, we want that the DNA fragment should be visible after electrophoresis. So we stain the DNA with some of the fluorescent dye or fluorescent molecule, and here we use ethidium bromide. So this ethidium bromide intercalate between the bases of nucleic acid and allows the convenient detection of DNA fragments. So under UV, this DNA plus ethidium bromide gives a reddish orange color which can be easily detected. And the sensitive
Ma'am.
detection, sorry, yes.
Yeah, ma'am, but the thing is that like DNA can be detected even without EDTA. Like when in water, something like DNA denaturation happens. At that time, we can detect the DNA even without using EDTA. So, like, why do we have to use EDTA?
Because we want
Like
prominent,
ethidium bromide.
so
Sorry.
Yes, I understood your question. So we want to see the we don't want to see the denaturated DNA. We want the intact DNA bands where it is coming. So actually, we use a ladder. We want to quantify the molecular size or the exact quantity of the DNA. For that, we want that the DNA should be intact and as well as the place can be found out. For that, we are using ethidium bromide. Can I answer your question?
Yes, ma'am. Yes, yes.
Okay, so the sensitive detection can go up to, like, it can measure at a very low concentration of 0.1 microgram of DNA. But the standard concentration which we use for ethidium bromide in electrophoresis is between 0.5 to 1 microgram per ml. Now, after the staining of dye, the UV absorption is measured, and the maxima can range at 300 and 360 nanometer, and emission maxima is at 590. And the detection of bounded DNA, that is ethidium bromide with the DNA, ranges from 0.5 to 5 nanogram per band. Now, alternative to ethidium bromide, we can also use other molecules like methylene blue, cybersaf, xylene cyanol, or bromophenol blue. Now, what is DNA ladder? I have been like, how will you quantify the DNA? Now, till now we have seen that the DNA fragments has been separated, but how will we know that what is the particular size is of the DNA? How many base pairs are present? So for that we use a DNA ladder. So what is DNA ladder? It consists of DNA of known concentration. So there are, it's a sample of DNA with fragments which we know the exact base pair size. So we run the ladder on one side and the unknown on the other, and from that we can compare what's the size of our target or unknown DNA. Ethidium bromide, as I have already mentioned, is a fluorescent dye. It just intercalate between the bases of the DNA and help us to localize where the DNA is present under UV light. But the problem with ethidium bromide is because it can intercalate with DNA, it can intercalate with the DNA of our body also. So, and it is a powerful mutagen. So, it can cause mutagenesis in our body. So, we should handle the gel and ethidium bromide very carefully, and we should wear gloves before doing this type of experiments. And after the experiment is over, the agarose gel with ethidium bromide should be disposed at hazardous waste very carefully. Now let's summarize the entire thing. What is agarose gel electrophoresis? So if I want to summarize everything, I will take only 3 steps to summarize this. So the first step is, first of all, we prepare a gel with a DNA dye, that is ethidium bromide. The agarose concentration is dependent upon the size of the fragment of DNA we want to separate. The second step is we load the DNA sample onto the gel and then we run the voltage at the optimum condition so that the separation is proper. At last, we visualize the gel in UV and images recorded. So here, the first step is given, preparation of agarose gel and casting of gel. So as you can see agarose in this picture, it's a white color powder. We add this agarose gel about 0.8% as I have already mentioned in 1x TAE buffer. Now we will see how the next steps are taking place. So we boil the agarose in 1x TAE buffer and make sure that all the crystals of the agarose is completely dissolved. And let the entire thing be cooled at like less than 50, and in the meantime we clear our casting trays and combs properly with ethanol so there is no contamination. We apply the comb in order to make the wells. Now we clamp everything properly so that the gel don't disperse. The formation of bubbles which you can see tells that their optimum temperature has been reached, and now we have to add ETBR, the staining dye. We will mix it properly and slowly pour it into the casting tray. After the gel is solidified, after 30 minutes, we will remove the comb very properly so that they don't dislocate, and then we will put it into the electrophoresis chamber. Now in the next step we will see how the loading of samples and ladder will take place, and then we will run the entire system at 70 volt for 1 hour. These are the samples of DNA which needed to be loaded. Now we will add the loading dye, that can be bromophenol blue or orange G, and we will mix them properly with the DNA. Now we will add the sample to the wells very properly so that they don't get punctured. Now we will add the ladder. Okay. Now if the volume of the DNA is very less, then another process is taken place. We will take a paraffin strip and we will add the DNA sample and loading that together on this paraffin, and then we will load the sample on the chair, and then we will also add the ladder. Now we will start the process and the flow of current will take place in the instrument. So after the run takes place, we will view the gel in UV trans illuminator and record the image. How the image will come? The results are shown here. The ladder is generally 10 kb kilobase pair, and here the plasmid DNA can be seen. This is the end of today's presentation. If you have any question, please ask. Hello.
Yes, ma'am. Ma'am, like in video, there was something added. I don't remember exactly.
Which slide?
Uh,
Can you please tell me?
no, in video.
In which slide? Which slides video you are not able to
Yeah,
understand?
just this video.
22th or 21st?
22nd.
Okay. Yeah, tell me what you are not
Last,
able
last,
to.
just last. Yeah, ma'am. So, what is parafilm strip? Can you please explain again?
Yeah, it's a strip which is it's a rubber kind of parafilm is used to wrap something. So when you will come into the lab, you can see we are generally using the paraffin strips to wrap the material so they don't get contaminated. It's a plain rubber strip. Okay, so in that we use this generally for mixing because the amount of DNA is very small, so we can't use vial because if we use vial, then some amount of the DNA can remain into the vial itself. So we don't want to waste a single point of DNA as the volume is very low. Therefore, we place the DNA into this parafilm along with the loading dye, and then we mix it properly and then put it directly into the wells. Okay.
Okay, ma'am. Okay. Ma'am, again, like 10 kb base DNA is loaded in that loading part. well.
Yes,
So, ma'am,
yes.
like, how do we ensure that that is 10 kb? Like 10 kb will be very, very small, right? It will be even like nanogram, even very less than that. So,
So
like,
we
how do
so we have pipettes for that. So we load 10 kb with the help of the pipettes into the well. We can do it like properly. We don't need to like worry about that thing.
okay, ma'am, okay, okay.
Any more question? If you don't have any more question, then I will stop sharing my screen and I'm uploading this entire PPT into your files section. Please go through it, and if you have any question after that, I'm like in 15-20 minutes I'm there. So please ask me.
Ma'am, like this for this experiment, we don't have like data. So, like, for other experiments we were doing. So, in this report will be just like description of all these things.
Yeah, exactly.
Ma'am, one more question, ma'am.
Yeah, sorry, yes, please.
Yeah, ma'am. You mentioned like this is used for DNA and protein. Like, can it be used for RNA?
Yes, depend. This DNA, for DNA we use agarose gel, right? And for RNA, for protein we use acrylamide. So for RNA, it depends up what is the fragment length and things, and then we will decide the gel type. The gel electrophoresis can also be used for RNA.
Okay, ma'am. Okay, okay.
And one more thing, that the RNA should be double stranded. Okay.
Okay, ma'am. But RNA is generally found in like single stranded, I guess. Yeah.
Yeah. So for functioning in gel electrophoresis, you need to have the RNA in a double-stranded form, so that the ETBR can intercalate between them. Not the staining won't be done, and we can't see under UV.
Yes, ma'am.